Today I had the opportunity to present the updated clinical and translational results of the NEOSTAR study, which evaluated neoadjuvant nivolumab or nivolumab plus ipilimumab for resectable non-small cell lung cancer patients in the neoadjuvant setting. The rationale for this phase 2 multi-arm study stems from the evidence that, unfortunately, more than 50% of patients with stage 1 to 3 resectable non-small cell lung cancer will relapse if treated with surgery alone. And perioperative chemotherapy, as a means to prevent tumor recurrence in this population of patients, only provides a 5% improvement in 5-year-old survival as compared to surgery alone. Immunotherapy is being tested in the neoadjuvant setting with the goal to induce an anti-tumor immune response and eradicate micrometastatic disease, so to prevent this tumor recurrence and potentially improve outcomes in this patient. For this trial, the primary endpoint was the major pathologic response, defined as less than or equal to 10% viable tumor cells in resected specimens following neoadjuvant nivolumab or nivolumab plus ipilimumab. The NEOSTAR was a multi-arm phase 2 study in which eligible patients with resectable stage 1 to 3A and 2 single-stage non-small cell lung cancer were randomized at a 1-to-1 ratio to a nivolumab monotherapy arm or a nivolumab plus ipilimumab arm. And after 3 to 6 weeks after the last dose of immunotherapy, patients underwent surgery. We assessed radiographic responses with CT scan and PET-CT scans, and we collected for biomarker analysis baseline tumor specimens, blood samples throughout and post-immunotherapy surgical specimens, as well as the stool samples. As part of the statistical design, if 6 or more MPRs were observed after each of the treatment arm, then the specific therapy would be considered promising for further evaluation. In the intention to treat population, the major pathologic response was 17% in the nivolumab arm and 33% in the nivolumab plus ipilimumab combination, and 7 patients in the nivolumab plus ipilimumab group achieved MPR, and therefore this regimen met the pre-specified efficacy endpoint for being considered promising for further evaluation. We also observed 6 complete pathologic responses after the combination neoadjuvant nivolumab plus ipilimumab as compared to only 2 complete pathologic responses with nivolumab monotherapy. And we saw that tumors treated with the combination therapy had less viable tumor cells as compared to nivolumab treated tumors. We also noticed that there was a positive correlation between overall radiographic responses and overall pathologic responses. The toxicity profile was overall acceptable. However, we're still learning with this trial and with other trials testing immunotherapy in the neoadjuvant setting how to administer these agents to a population of patients with resectable disease. And we're learning about the surgical outcomes with these therapies. Preliminary results of biomarker analysis show a positive correlation between elevated baseline level of PD-L1 and radiographic responses, major pathologic responses, and greater tumor pathologic regression. We also saw from evaluation of surgical samples that nivolumab plus ipilimumab was associated with increased T cell infiltration and with particular subset of T cells that might be potentially reactive to the tumor and be responsible for an anti-tumor immune response.
So how do you think that these results will change or influence clinical practice?
Yeah, so this is a very important question. I think the fact that the combination therapy met the pre-specified trial endpoint to be considered promising tells us that maybe this combination deserves further evaluation, most likely in association with other therapies, chemotherapy or other immune checkpoint inhibitors. This can potentially affect in how many patients with resectable non-small cell lung cancer we can limit tumor recurrence.